Over the last few years, we have been proposing that we could treat tumors across the different types with the same therapy for as long as these tumors have a gene driver that is common, right? So we have done some studies to prove the point, like with BRAF and with NTRK and other targets. And the one that one of our investigators is presenting here, her name is Komal Javeri, and I'm the senior author, is that we are presenting the results of a basket trial for tumors that harbor PI3 kinase alpha alterations. And what we have observed is that there is a variety of tumors, and we're talking about head and neck and others, that they harbor these driver mutations in PI3 kinase, or they have amplifications of the PI3 kinase gene, and they are quite sensitive to these therapies. So we are quite excited. And since PI3 kinase alpha mutations and amplifications occur in multiple tumor types, so most studies are being focused in breast cancer, and rightly so, but you're going to have a whole set of tumors that harbor these alterations. And we will show actually quite nice clinical activity and beautiful water plots showing that this is working. Well, I think for the future of the patient, I think that we'll need to now is decide what do we do next, right? And I would like very much to have the possibility to explore registration studies in not just breast, but in other tumor types as well. And that's the implication. So we are presenting here, I'm just present one of the authors on beautiful work that is showing that we can detect, using good technology, next-gen sequencing technology, that we go deep and that we can incorporate a number of assays, that it's not just the classical NGS, but also methylation patterns and other copy number alteration, that we can really detect the presence of tumors by cell-free DNA. And I think that the implication of that is that we could begin to visualize the possibility of using this technology to diagnose cancer earlier. So this technology could be a phenomenal tool to screen populations at risk and to have a shift in the diagnosis from advanced disease to earlier disease, and therefore increasing the cure rates. And then also you could use this technology in these tumor types to monitor response to therapy. So the beauty of this is that you can repeat it, so you can look it in multiple times, and I think that that's also quite exciting. Much more convenient to patients. I mean, instead of having imaging studies that are not sensitive enough to pick it up. I mean, the most important thing is that this technology is going to be far more sensitive than imaging. But on top of that, it's clearly far more convenient to patients than just to have CAT scans or MRIs. So we're still going to be doing all these imaging tests, but I'm assuming they're going to be far less frequent than what we do now. And then the third implication of all this is that these cell-free DNA assays are helping us identify also what therapies to choose, because they are giving us genomic information about some of the key driver mutations that are present in the tumor. So it's not only telling us, the tumor is growing, but rather, actually I should say at the same time, it's telling us, well, and this is the way you should target the tumor because you have these mutations that are indicative of a dependency.